to HORIBA, the introduction of morphology and our quality slide programme. Today we're here with Andrew Fisher, our technical specialist, and myself, Anna Ellis. We're here to talk about the different cell categories and cell morphology, and we've got some really exciting news that we'd like to share with you before our Q&A session. If you have any questions during the webinar, please put them into the chat and we'll respond to them for you later. Thank you. Andrew, could you actually give me an example of what is cell morphology?
Cell morphology is pivotal in haematology, or the study of haematology, because in morphology we look at red cells which take oxygen to your muscles and CO2 back to your lungs, and then we also look at white cells, and there are 5 main types of white cells. There are neutrophils, lymphocytes, monocytes, eosinophils, and finally basophils, which are very rare even in normal patients, so that's a basophil. And you can see from here there are substantial differences between the 5 main types of white cells. So when a user is looking down a microscope at a blood film, they can see these cells, and then they will do what is known as a manual differential, where they look at a minimum of 100 white cells and categorise them into neutrophils, lymphocytes, monocytes, eosinophils and basophils, and also look at the red cells. And in so doing so, they can then give a definitive diagnosis for that patient. It could be something as simple as iron deficiency, or something as more complicated as a leukemia.
Thank you, Andy. Could you provide me with some basic characteristics of the white cells, please?
Yeah, sure, Anna. Using HORIBA QSP program, I can quite easily show you different white cells and what they look like in great detail. So now we're looking at a neutrophil, and we can see a neutrophil is quite a large cell as compared to the red cells. Its nucleus is multi-segmented, and we can see granules, and these are primary granules or azurophilic granules. Neutrophils, they're the commonest type of white cell in a normal adult blood film. Next, we have lymphocytes. So lymphocytes produce antibodies, helping your immune response, as do all of the white cells. And you can see here the big difference between neutrophils and lymphocytes is that the lymphocyte has a single nucleus, it's not segmented, and there's not much cytoplasm as compared to the nucleus. Also, a good little tip is that the nucleus of the lymphocyte is, generally speaking, about the same size as a normal red cell. So when you're looking down a microscope, you can work out whether the red cells are normal in size or large, i.e. macrocytic, or microcytic, which is small. So that's a lymphocyte, very important cell, as all the white cells are. Now we have monocytes. So these are large cells, and as you can see, we've got a lot of cytoplasm there, and this colour of the cytoplasm is often called a ground glass appearance. The nucleus is quite convoluted. You can see in the cytoplasm, we can see it's vacuolated. Quite large cells. Monocytes are quite interesting, very interesting in fact, because these can transform into macrophages and also dendritic cells, which help in antigen presenting. So the monocyte will go into the dendritic cell, which then processes the antigen so we can produce an antibody, and then the lymphocytes will produce the antibody. Eosinophils, I think these are quite pretty cells. Bilobe nucleus, very bright granules. So eosinophils, you'll see in eosinophilia in things like hay fever, so you see a lot of eosinophilias in summer, and we can see they're easily contrasted against a neutrophil because they're bilobed and because of the appearance of the granules. And last but not least, we have basophils, which are the least common of white cells. So the basophil is quite similar to an eosinophil in the fact that it's bilobed, but the major difference is these granules are very dark, and they're often so dark that they can occlude the nucleus. So you're lucky to see 1 basophil per 100 white cells. So when the morphologist is looking at the blood film, they're doing a differential, counting the number of different white cells. And obviously there's more white cells than this. We have precursors of these, and we're also looking for things like blast cells, which will be an indicator of leukemia. And then we will also look at the red cell field to see about the colour of the red cells, the size of the red cells, the difference in shape of the red cells. And again, what we can do after looking at blood film is we can give a definitive diagnosis.
Thank you, Andy. Andy, can I just ask you, what software are you using?
Okay, I'm using the HORIBA Medical Quality Slide Program software, which is a digitised slide from a patient, and it's been digitised, and the differential is being performed by our morphology expert over in France, and then is available to all our QSP users.
So how long does it normally take to learn blood films and blood morphology?
You never really stop learning. There's always something new. The biggest problem with inexperienced morphologists is they need to know what the difference is normal is. There's no such thing as a typical neutrophil. All neutrophils in a normal adult have different variations. So this is from a normal patient. This isn't our normal patient. And we can see, just looking through neutrophils, we can see a wide variation in number of segments, degree of granularity, and that's the same for lymphocytes, monocytes, eosinophils and basophils. So that for the trainee morphologist, the first thing they've got to learn is what is normal.
And it's my understanding that currently within universities it's only a very small section of the courses dedicated to morphology, and when the students appear in the lab they have to use a twin-headed microscope. Would this software assist with training and competency?
Oh, definitely, because with this software we can now have a 10 user license, which means you can have 10 separate PCs set up, so 10 concurrent users. So that will mean in a university laboratory environment you could have 10 people looking at the same blood film, and then from that you could produce a training package and also a competency assessment package, so you know that person knows what a neutrophil is, a lymphocyte is, and there's no ambiguity. They have to look at all the white cells, they have to differentiate all the white cells, so it would be a big leap, so that when these students then go to work in a haematology department, there'll be some proof that they know what morphology is and a level of competency.
Thank you. Is it possible to look at the programme?
Yeah, it sure is. It's a very easy and simple thing to use.
We're going to spend a little time now talking about the Quality Slide programme and how it can help you in training your new morphologists. The beauty of the QSP programme is that you can have unlimited number of users, so no matter how large or how small your class is, you can add the relevant number of users. So let's log in. We've developed a specific slide where every cell has been annotated to what that cell is, and also an interesting fact about those cells. So let's take a look. So it's in the training package and it's slide number 3. When it first opens up, the first thing you'll see is all the white cells, and they've all got a blue box around them. That means that we've attached a comment to this particular cell. So for example, the cell comment is it's a neutrophil, and a little fact, in this case, note the fine granules in the cytoplasm. If we double click on the cell, it magnifies that cell so we can see it in greater detail. Here we can see what appears to be a drumstick appendage, which is present in 10 to 15% of all neutrophils in females. So we can go along double clicking if we want and seeing what the cell is, or it's probably easier just to hang over the cell with the cursor and we can see, for example, that's a lymphocyte. Neutrophil. Here's a eosinophil, and we can see it's very much different from a neutrophil. The granules are large and eosinophilic, and the nucleus is only bilobed rather than multi-segmented. Whereas if we look at a neutrophil, we can see it's totally different. If we look at a monocyte, we can see that the cell is very large, The cytoplasm often contains vacuoles, and the nucleus is quite convoluted and complex, and it's one of the largest white cells that is found. We can see the size compared to the red cells. So if we want to, we could just stroll through, noting what each cell is, why it is that particular cell. For example, why is that a monocyte? It's a monocyte because it's quite large. The colour of the cytoplasm is different to a lymphocyte and a and a neutrophil, and the nucleus is quite large and convoluted. A neutrophil, another neutrophil, and as we're going through looking at the neutrophils, we can also see that there is a natural variation from one neutrophil to another in degrees of granulation and degree of segmentation or lobulation. Here's a basophil. You see probably 1% of all normal white cells are basophils, and we can see the very large granules, and you can just about make out the nucleus. The granules often occlude the nucleus, so you can't get a good sighting of it. So using this tool, this QSP slide, we can instruct students in what each normal white cell is, and hopefully they can then understand what makes a neutrophil, a lymphocyte, etc. If you want to test them, we could ask them to perform a differential. What we would do is we may show them this slide and then get them to do a differential on another normal slide to see if they've remembered the difference between neutrophils, lymphocytes, monocytes, eosinophils and basophils. Now I'm going to show you how QSP can be used to assess competency in users. So if we go to train diagnosis, pick this package and we'll pick this slide. This happens to be a normal slide because what we want to do is show you how the previous slide with all the cells annotated can help you do some basic morphology training and then check that training is being taken on board by the student. So you can see here, these cells don't have any blue borders, they have no comments. We can double click on the cell to magnify it, and currently this cell is not being classified. So our job is to classify these cells to differential in effect. So if we pick a cell, we can place it into its relevant classification. If we want to, we can pick multiple cells by selecting the cell and holding the control key down while selecting the cell. And here I can go along selecting what I think are all one type of cell. And I happen to think that these cells are neutrophils. So we can then drag and drop all of those cells across to the neutrophil classification. And we'll see over here that we've classified 18 cells out of a total of 105. We have to classify every cell, we can't ignore a cell. So we'll carry on classifying cells, holding the control key down, selecting the same cells of the same classification. As well as dragging and dropping those into the classification, we can also use these shortcut keys. So if I press Z, you'll see that number increase from 18, and we've just added another 10. Let's choose some cells of a different classification, and I am going to make some mistakes because I'm an inexperienced morphologist and these cells look all very similar to me. So I will make some mistakes as I'm going along. So these I think are all lymphocytes. So if I press X, we can see that the cells go over as lymphocytes. So I've got 48 out of 105. Let's pick these cells, these rather colourful cells. We think these are in eosinophils. Oh, there's one there. We think these are eosinophils, so we press the V. So we very quickly can classify cells. So we're doing what's known as a white cell differential. So we're classifying each cell into a classification. The number of cells of lymphocytes, neutrophils, monocytes, etc. is useful. If it's abnormal, that obviously says that the patient may have a problem. If there's too many lymphocytes, for example, they may have a disease called chronic lymphocytic leukemia. If they've got too many neutrophils, it could be because they've got an infection and the neutrophil response has been invoked, and we've increased the number of neutrophils within the blood. So you can see it's very easy to do. I'm just selecting multiple cells and then placing them in the classification. So we'll call those lymphocytes. So I've finished. We can now look at the red cell field, and we can see a red cell field. We can zoom in, we can walk around, have a look. Included in some of the slides we have things like malarial parasites, sickle cells, red cell inclusions like Howell Jolly bodies. So we do have a number of red cell abnormalities included within the slides, and the user can then add these morphological comments should they require. If you're just interested in white cells though, you can simply click on the finish, and that now means that that slide has been completed, and this user can no longer see that slide, do that slide again. What they can do though is to see how well they've done. So if I go to evaluation and report, pick the slide that I've done, it has to be number 4, and we can see immediately what this user has done wrong. We can see we have every cell displayed, and if we've classified that correctly, there is a green border around the cell. If you classified it incorrectly, we see a red border, and if we select the cell, we can see that its true class is a monocyte, and we called it a lymphocyte. So we can see we're making a common mistake. We're making the same mistake over and over again. We're calling monocytes lymphocytes. As well as seeing it on the screen, we can produce a report. So if we create a report, and we'll create a PDF, and the PDF will be displayed. From this PDF we get a breakdown of the results. So we can see here a breakdown of the results. The number of cells that the expert or the reference saw, which is in green, and the number of cells of that type that this particular assistant saw and counted. And we can see that they didn't count any monocytes, whereas the expert counted 5. And we counted 5 more lymphocytes. If we go down the page, we get it in numerical data, and we get some data about sensitivity and precision. So from that, we could use that to calculate uncertainty of measurement. But the biggest uncertainty of measurement in blood films is, how do you know that you all look at the same white cells? In QSP, we know everybody's looked at the same white cells, and they have to classify every cell. Here we have an area where we can see a picture of the cell that we've misclassified, what we classified it as, and what it should be. So within QSP we get some reflective learning where we can see what we've done wrong, and then we can then act on that accordingly.
Thank you, Andy, that was really informative. Is there any extra literature or information that accompanies QSP?
Well, yes, there is actually. We do a monthly QSP newsletter, and we also have available some nice wall charts detailing hematopoiesis and erythropoiesis. Further details will be available after the Q&A session.
Do your slides cover a range of abnormalities?
Yes, indeed. In fact, I've got 7 years worth of different slides, and if necessary I can produce what we call a package, which includes slides from various abnormalities within the same group. So this one, for example, it's all to do with very abnormal white cells. So we've got an AML with blast cells, maybe some hour rods, and if required by a customer, we can create actually a specific package for specific abnormalities, such as red cell abnormalities or in this case, white cell abnormalities.
So are you able to create a package then that's suitable, say, for a basic user, somebody that's only just learning about morphology, to a more senior expert for, say, consultant level and things like that?
Yeah, that's entirely possible. We have a vast number of normal slides, slightly abnormal slides, and quite esoteric slides, so we could break that down into different levels of competency if that's what's required. I already have a level 1 competency, which is just normal slides, a level 2 competency, which could be somebody who's just learning morphology and you might want them to work on their own at night, and that could include things like a malarial parasite, a blast cell, or low platelets.
Thank you, Andy.
Thank you.
So thank you for all your questions so far, everybody. If you'd like to put any more questions into the chat box, that'll be excellent and we'll answer them later. But Andy, I'm really excited to find out what the new news is with regards to QSP.
Okay, yeah, well, Anna, it's literally hot off the press that QSP v2, if you have a site license, you can now use that site license through a VPN, a virtual private network, which means that you don't have to be on that particular site's network, so you can be almost remote accessing into QSP. So that will enable more users to be able to connect using the site license. And another thing I'd like to mention is that the newsletter is now 1 year old.
Wow, that's amazing.
Yeah, so it's been 12 months, 12 difficult months for everybody due to certain things like COVID. And it's just showing HORIBA's commitment to supporting the scientists out there doing good work in the field, and those who are particularly interested in blood film morphology. So the QSP newsletter, for those that don't know, it's regarding blood morphology, and we have a case study, and the recent case studies have been things on like hereditary spherocytosis, thrombotic thrombocytopenia purpura, myeloproliferative disorders, and there's also a fun quiz to do every month that we publish on social media. So yeah, the QSP newsletter is edited by a small team of Kelly Duffy and Shuban, and I'd like to thank those for all the contributions to enable the ongoing success of the QSP newsletter.
No, that's brilliant. And for everybody that would like to receive the QSP newsletter and some cell posters, along with any additional information, we are going to be sending an email out after this with the links on there, so you can access that and sign up to that. But now I've noticed we've got some questions, so if that's okay, we'll just start with the questions. And again, please add some more in there. The first one, I think it's quite relevant as well, Andy, is what changes might you see in blood morphology in a patient that's been hospitalised with COVID-19?
Yeah, well, there's quite a lot of information out there at the moment, particularly on patients that are unfortunately having to be ventilated on ITU. As we know, ITU patients, any patient who's on ITU, critically ill, a lot of interventions, therefore blood film morphology is affected in respect of, you often see neutrophil toxic granulation, etc. It's been noted in some COVID patients that the neutrophils are containing dull bodies or dull-like bodies, and there's some literature about that. As well, you can sometimes see abnormalities in lymphocytes, apoptic lymphocytes. So a general picture of a really aggressive infection plus being within ITU. So there's quite a few papers out there that we can all look at.
And what are the differences between the adult and pediatric blood morphology?
Yeah, well, pediatric morphology is, for you to look at a particularly premature baby, if you looked at it not knowing it was a premature baby, you'd be quite worried because it's very abnormal, nothing like an adult blood film. So you see changes in neutrophils, monocytes, lymphocytes, sometimes even occasional nucleated red cells. And it's something that we have had slides in the past where we've had premature babies included in QSP packages, and it's something that we could maybe do in the future as well.
And do you feel that this package would be suitable for biomedical students in the university setting?
I do, particularly now we've got the VPN connection, and which will assist universities in remote working. So I've only just started thinking about the VPN connection and really how it can be used. So it could allow remote access into a central QSP server, for want of a better word. The packages themselves, as I mentioned previously, we can tailor that to either be all normal or have red cell packages for things like sickle cell, owl jolly bodies, and also quite esoteric white cell abnormalities, including some of the leukemias, hairy cell leukemia, and things like May-Hegland anomaly. So yeah, I think it can be of use within a university environment. In fact, we have put it into a couple of universities in the past where they had specific morphology sessions, and we placed that the QSP. It was a physical QSP, but now we can go VPN, so it should be much more easy for us to do.
Okay, sorry, I'm trying to read everything as quite quickly as they're coming in there as well. So there's another question on here saying the application of the package could be useful in healthcare training scientists on their pathways as well.
Oh, definitely, yeah. I mean, that's where the main users of the system are in laboratories where you want to prove competency. Competency is a very difficult thing to prove in morphology, because if you give someone a blood film, they can claim not to have seen a particular cell. Whereas if you give them QSP to do, you can definitively, because I have to look at every cell and I have to categorize every cell, you can definitively say and comment on their competency. So it's a great way of ensuring competency, and it's something that's not threatening. And the uptake is pretty good, and the users tend to see it as a positive thing rather than a negative thing. So and it's instructional, because at the end of the report you do get some reflective learning where you can see the cell that you got wrong and it tells you what it should be. So therefore you can say, oh yeah, I can see that now. So yeah, it's definitely of use for competency assessment.
Can you also tell me, can we download the slides and store the slides and use them at a later time? That was another one.
You can download on the website. We keep this month's slide package plus the 2 previous month's slide packages. So we've got you can in effect download 3 months worth of slide packages at any one time. And if you want a specific slide, then if you make inquiries, then there's a possibility we could produce that slide just for yourselves.
So are these packages only available to existing HORIBA customers, or?
Oh goodness, no. We market the QSP software to anybody. You don't have to be a HORIBA user. It's nice if you were, obviously, but yeah, we will sell it because it's a standalone package. So yeah, anyone can buy it.
Okay. Will the QSP be integrated into the P8000 software at this precise moment in time?
So yeah, okay. So thanks, Dom. Currently, it won't be. As you know, we have an interface to Television, which is an interactive interface, but it will stay as a standalone product because we do sell it to non-HORIBA users.
Brilliant. And do the packages include all cells characteristics descriptions for the training purposes, or is it something that we can create and provide to everybody, or make it more bespoke, like you were saying before?
Yeah, that package that was illustrated is one that I created not just for this event, but I've created it in the past. It's, as you can imagine, quite time consuming. It's theoretically possible I can create slide packages with cells annotated that include things like leukemias, so you can point out the fact that there's hour rods present. You could point out the fact that there's dull bodies, Pelger-Huey cells. So yeah, we can create an annotated package, but it's not something that can be done quickly. And we would do our best endeavour to do that in a reasonable timescale, should it be required.
And to work with the customers.
Oh yeah,
build up what they require on there, of course. Yeah, definitely. I think we've covered that one, but the application of the package would be useful in healthcare and scientists. I think we've covered that one already, Andy. I do apologise.
Okay.
How is the atopic cells looking like, especially neutrophils and monocytes?
Sorry, what's that one?
How's it atopic?
Yeah, apoptic cells, they are cells that are reaching the end of their life, so you don't normally see them in a normal patient, but in COVID patients particularly, you can see apoptic forms. Neutrophils, the nucleus, will be quite extended, and the granulation will have gone, and they just look. You see that the neutrophils, but they look very, very abnormal. Apoptic lymphocytes tend to coalesce, they get pycnotic. You can tell that the lymphocytes, but you see that things aren't correct. The nucleus will be breaking down, etc. So I'm sure if you Google that, you will see some good images.
Mm-hmm. And if somebody was coordinating training, what would they be able to review the staff performance? Would they be able to review the staff performance using the QSP?
I don't think I showed you that.
No.
But there is a specific report called the expert report. I don't like the word expert, but it's an expert report. And on that, you can see the performance of every user for each cell line. So you can see that they've counted all the same neutrophils as the reference. So it's a one report where you can see everybody and their performance. So it's fairly quick to do. You can also download it to an Excel spreadsheet should you want as well, and display it graphically.
Okay, no, that's brilliant. And is there any other options of reading that you might advise for people that was interested in particular morphology?
Yeah, well, personally speaking, I don't think you can go far wrong with something like a book called from Barbara Bain. Barbara Bain is a world-renowned expert on morphology, so any book by Barbara Bain would be pretty good. One thing we do have, if you go onto our HORIBA website, at the bottom of the page, there's a link to an atlas called Haematovision, and on that there is a cell atlas detailing cells, what they can be confused with, as well as I think it's 9 clinical studies at the end. So that's, if you go onto a HORIBA web page, drop down, and you'll see the Haematovision website, and it's free, and you can also have it on your iPhone.
It is an excellent tool, definitely. And if anybody's interested in these packages, the best course of action is to respond to the email that we're going to be following up on there. So if you're interested sort of in the price, any further details like that, we are doing a follow-up email, or please contact us directly that we can come back to you on there. Sorry, I'm still trying to if the QSP user has an interest in blood film from the lab, could this be turned into a digitalized slide?
Yes, in fact, we do have a specific form that we require to be filled in, and we need to send it over to Montpellier, the south of France, where they will digitize it. So we actually need preferably 2 slides, just so we have sufficient cells to digitize. So yeah, and if anyone's got an interest in slides, they can email us and we will send the relevant form out.
Okay. We've kind of touched on this already, but if anybody require any sort of bespoke packages to be built, we can do that still for them. If they wanted sort of from a basic to a more expert, or sort of the sickle cells or malaria, like I've already mentioned, we could create that and build that.
But it's not something we can do straight away.
No.
It is quite time consuming to do.
What career paths involve looking at morphology?
Well, a biomedical scientist, particularly in haematology.
How do you see this being used aligned with clinical teams and laboratory teams in accordance to the morphology results?
Yeah, consultant hematology, it's a strange thing, but it's a fact of life that in most hospitals, if you see an abnormal blood film, you'll refer it to a consultant or a registrar. You don't see the consultants as registrars daily. So on a few of our QSP customers, they actively involve the consultants and registrars. They use them as a user to check as well. So and in fact, one site, they actually purchased the QSP so they could train their registrars in morphology. So it can be used as a training tool, and also to assess the competency of junior members of the medical staff. It's a good thing to get the consultants involved. If you get the consultants backing, you will get the money for the QSP product. So it's nice to get them involved right from scratch. But one thing I can say is that it isn't a clinical interpretation package, it is a morphology package. So although there is clinical data there, it's not something where we're asking for somebody to put a definitive clinical diagnosis in.
No, and I think it covers a lot of different aspects, both from sort of the universities, from the basic level all the way up within the in the NHS sector, and it can provide a great grounding for everybody, like you've just mentioned, and it's a brilliant learning consultative tool that we can take on board.
It's very easy to use. It's non-threatening. I used to work in the labs, and whenever we got the Nequast blood films, everybody left the room because it's felt as quite threatening. Whereas with QSP, it's very intuitive, and I think it's the way laboratories are going, haematology laboratories are going, with the advent of more and more digital morphology systems. This is the way biomedical scientists are going to be looking at cells digitally. They're not necessarily going to be looking down the microscope all the time. So I think it's up to date, it's easy to use, informative, and non-threatening. So it does get used in places where the board purchase it.
And do you feel that morphology is a dying art? From what I've seen, it seems to appear like that, but what's your thoughts on there?
Well, it cannot be a dying art because we need these people to be able to look at blood films. It is an art that is not being forgotten about, but it is an art that is more and more difficult to train because there's less and less time in labs for people to sit back and train. So QSP will ensure it doesn't become a dying art because it can't be a dying art. You will always need to look at blood films to give a definitive diagnosis.
Brilliant. I think that's all the questions that we've had come in today. So if anybody does have any other questions or any queries, again, please feel free to email us. We will be following it up with a follow-up email, which will again provide you with the information for the QSP website, the newsletter, the quizzes, along with the academic website as well, which has a more plethora of webinars on there and training tools that we're very interested. So we'll be adding that link on there. And we've had a few queries with regards to costs as well, so we're quite happy to contact you direct and discuss that further with you, because again, each different package in different universities and NHS sites need different variants on there. So we're very happy to follow that up for you. Thank you.
Okay, thank you very much.
Thank you.
Thank you for your time, everybody. And
thank you.
Keep safe.